Hello everyone, welcome back to our online lecture series. So in today's lecture we will be learning about the information transfer purpose. So here we will be learning about the purpose of information transfer in biological systems. So here the information means that is the genetic information. So when we were studying the biomolecules, there we have learned that these DNA and RNA are the biological molecules which will carry the genetic information. So and we also studied that the genetic information will be transferred from one generation to the another generation or to the offspring with the help of DNA. So how this DNA will transfer the genetic information from parents to the offspring, so that we will be learning in the present lecture. So here if you see the central dogma of the DNA information transfer, so you can see that here is the DNA and it will transfer the information, genetic information, through the transcription process to the RNA, and this RNA will translate the information to the protein. So that means this RNA will synthesize the specific protein, and these proteins will make the offsprings or traits or the next generation kids. So this is all about the central dogma of the genetic information transfer. But if you see here, this how does this genes code for the proteins? So how these specific genes on the DNA will synthesize the specific protein? So here you can see, so particular gene will synthesize particular genetic information that is present on the DNA will synthesize particular proteins, and these proteins will make the cells, and these cells will combinedly make the babies or the offsprings. But how this information will be transferred, how these genes or the information on the DNA will be utilized for the synthesis of proteins. So here you can see the picture that so like whenever we make any cookies or any food items, what we will do, just we will follow the recipe. So whatever the recipe says, that whatever is written in the recipe is to add some ingredients in that to make this particular item. So like that way we will follow the recipe and we will make the cookies. So in the same way, these proteins also synthesized by the by following the information that is present on the gene. So this gene will be acting as a recipe for the preparation of protein or for the synthesis of protein. So this is all about the genetic information transfer, which is that is what we call the gene expression. Here means the gene is expressing its characteristics in the form of protein. So that is why, so whatever the physical appearance that we can see in the babies or this offspring, so that expression, that is nothing but the expression of gene that is present in the embryos. So that is what we call it as the gene expression. So how this gene expression takes place, so that we will see now. So during our biomolecules lecture, we studied that DNA is a genetic material, right? So and it carries the genetic information from one generation to another generation. So these are all the things that we have studied in biomolecules lecture. So now we will see actually how this information exists in DNA. Basically how this information, whatever we are talking about the genetic information, how this genetic information exists in DNA in the first place. So to understand this, first we need to have a clear idea about the genetic code. So what is this genetic code? First look at the structure of DNA molecule. So here we have already studied the DNA molecule is composed of three kind of components like phosphoric acid, deoxyribose sugar, and along with these two things, the nitrogen bases will be there in the DNA. So there are four types of nitrogen bases that also we have studied, that adenine, guanine, cytosine, and thymine. So these are the four nitrogen bases that are present in DNA. In case of RNA, this thymine will be replaced by the uracil. But out of these three components like phosphoric acid, deoxyribose sugar, and nitrogen base, sugar and phosphate forms the backbone of the DNA, and they are always same. There won't be any difference in the two components, in these two components throughout the DNA molecule, right? So these molecules cannot carry the genetic information because they are not varied. Like if you take any gene sequence, so everywhere this ribose sugar and phosphoric acid will be same. In fact, you take any individual, there will be a change in the sequence, but there won't be any change in the structure or sequence of the phosphoric acid and deoxyribose sugar, because they are forming the backbone. So they are not varied from one individual to another individual or from one gene to another gene. So because of that, but we know that the genetic information varies from one person to other person. So in that case, this phosphoric acid and deoxyribose sugar, these two things can never carry the genetic information which is varied, because these two things are not varied. These two are same in every individual and every gene, so they cannot carry the genetic information, right? Then who will carry this genetic information? So out of these three components, only the nitrogen bases vary from one segment to another segment. And in fact, if you see the nucleosides also, every nucleoside will have different, some nucleosides will have adenine nitrogen base and some nucleosides will have thymine and all these kind of things. So because of that reason, this genetic information depends on the nitrogen base sequence, because if you see the sequence also in the genes, so the nitrogen bases will not follow the same sequence everywhere, so the sequence will change. So because of that reason, the genetic information depends on the sequence of the nitrogen bases, not on the basis of the phosphoric acid or deoxyribose, because they are not carrying the genetic information. The genetic information will be carried only by the nitrogen bases. So these are four nitrogen bases like adenine, guanine, cytosine and thymine. So these four nitrogen bases are considered as four alphabets of DNA molecule. So these four alphabets of DNA molecule, like all these nitrogen bases, this will be used to write the genetic code. So that based on the genetic codes, they will synthesize different proteins. So George Gamow was the first person who has proposed the basic structural unit of the genetic code. So the basic problem of this genetic code is to indicate how information written in 4-letter language of nitrogen bases of DNA can be translated into 20-letter language of the amino acids proteins, because we have already seen that there are 20 amino acids. So these 4-letter code has to synthesize 20 different amino acids. If each letter will synthesize one amino acid, they can synthesize only 4 different amino acids. But we know that we need 20 different amino acids. So how this 4-letter code is going to synthesize 20 different amino acids? So that here we are talking about the genetic code or codon. So basically, what is that code or codon? So this is nothing but a nucleotide that specifies the amino acid. So each nucleotide, or the group of nucleotides that specifies the specific amino acid, that is what we call the codon. So the simplest possible way or the code is a single code system. So if each one nucleotide code for one amino acid, but in that way what it can do maximum code for the 4 amino acid, because we have only 4 types of nucleotides already we have discussed. So in that way it cannot code for the 20 amino acids. And the second possibility is that just take the 2-letter code. So in that, what will happen if you do the permutation combinations with two letters of these 4 letters, so maximum they can form the 16 codes. So that means they can code for 16 amino acids. That is also not sufficient, because we need 20 amino acids. So this 2-letter code is coding only for the 16 amino acids. So then the alternate way is the triple code, or that is what we call the triplet code. First one is the singlet, that was not sufficient to code for 20 amino acids, and second one is the doublet code, that is also not sufficient, and the last one is the triplet code. So if you take 3 letters or 3 nucleotides together, three nucleotide letters together, so in that way it can form the 64 codes by using the permutation and combination methods. So these 64 triplets code can easily code for the 20 amino acids. So that is why they considered it as the basic and the best model for the coding of amino acids. So here on the right side you can see the genetic code table. So you can easily draw this table just by taking the 4 nucleotides on the left side and 4 nucleotides on the top and 4 nucleotides on the right side. Just I am explaining you one first table, that on the left side it is uracil, and on the top it is uracil, and on the right side uracil, cytosine, adenine and guanine. So you take one letter from each side. So the first one, if you see, it is taking U from the left and U from the top and U from the right side. So that is why it is forming the U. And the second one, UUC. So here it is taking U from the left and U from the right, and from the sorry, U from the left and U from the top and C from the right side. So that is why it is forming the UUC. So in the same way, the third one UUA. So it is taking U from the left and from the top, and it is taking the A from the right side. So here you can see that there are 64 codons. Each codon will be having three letters. So here we have total 64 codons, but we have only 20 amino acids, right? Then what about the other codons, what they will be doing? So what they will be coding for? So here you can see that beside the few codons it is written as the start. So that means here I have not mentioned the start, but here you can see on the left side, the third box, it is written the methionine, MET. So this is nothing but the start codon. So whenever the ribosome find these codons, they will start the protein synthesis. That is why we call them as the start codon. And beside the few codons, it is written as the stop. You can see that is written in the red color on the right side top. So that means these are the stop codons. So whenever the ribosomes find the these codons in a sequence, then they will stop the protein synthesis, right? But still the number does not reach to the 64 because we have only 20 amino acids, and some codons will code for the stop and start of the protein synthesis. But what about the other remaining? One thing you should remember here, that some amino acids are coded by more than one codon. So here if you see the example on the top, it is written UUE, UUU and UUC. These two codons will code for only one type of amino acid, that is phenylalanine, and below that it is written UUA and UUG. So these codons will code only for leucine. So whenever the ribosomes will find the codon UUA or UUG, so they will synthesize same kind of protein with this, they will attach the same kind of amino acid, that is leucine. So in the same way, some of the amino acids are coded by more than one codon. So for this reason we call that genetic code is degenerated. That means one amino acid is coded by more than one codon. So for better understanding, you can look at the example that I had given at the bottom of the slide. So here is the sequence of mRNA. So whenever this mRNA will pass through the ribosomes, so whenever the ribosomes find the code, that is AUG, which is written as the start codon, so they will start the protein synthesis. So AUG codes for the methionine that already I explained at the top in the box. So whenever they will find the AUG code, so they will start linking the methionine here, and then AUC, which codes for the isoleucine, and UCG, which codes for the serine. So based on the sequence that is present on the mRNA, these ribosomes will start incorporating all these amino acids into the polypeptide chain. So in the same way, when they will find the codon UAA, so that codes for the stop codon. So whenever they will find the stop codon, so they will stop the polypeptide synthesis. So here you can see UAA is coded for the stopping of protein synthesis. So because that is why we call it as stop codon. But one thing just you remember, that during the protein synthesis, these ribosomes will not synthesize the amino acids. Just what they will do, just they will synthesize the proteins by using the amino acids as the building blocks. So they will use the amino acids as a building block, and then they will synthesize the protein. So just they will be attaching the amino acids to the polypeptide chain based on the genetic code, but they will not be synthesizing all these amino acids, but they will synthesize the proteins by attaching the amino acids and forming the polypeptide chain. Now we have seen the concept of genetic code and how many nucleotides will code for one amino acid. So these are all the things that we have already we discussed, and now we will see that what is the process of making a protein by using the genetic information that is present on the DNA. Synthesis of proteins by using the genetic information that is present in DNA will take place in two steps. So first step is called the transcription, and second one is the translation. By using these two steps, it will transfer the genetic information, or it will copy the genetic information. It will make the proteins by using the genetic code that is present on the DNA. In this central dogma of biology, that the genetic information, that DNA is transferring the information from DNA to mRNA. So it is just copying the sequence from DNA, and it is forming the mRNA. So this process is called the transcription. So now the genetic code is copied to the mRNA, and this genetic information will go to the ribosomes, and there in translation process what it does this, and ribosomes will synthesize different types of proteins by using the genetic information that has been copied from DNA to mRNA. By using this genetic information they will synthesize different proteins. So this is what we call the translation process. Here in this picture also you can see in detail transcription and translation process. So in the first case what is happening, the information is being copied on the RNA. So here you can see already we know that adenine will bind with the thymine and uracil, guanine will bind with the cytosine. So these are the things because here you see second place it is adenine, but in whatever the information that is being copied, that is being copied to the RNA, but in RNA we don't have the thymine, so that is why in place of thymine this adenine is binding with the uracil. So that also we already discussed. So this, by copying this information, it is making the complementary RNA. So this RNA is forming the transcript. So this transcript or this mRNA will be utilized for the synthesis of polypeptide or protein in the next translation process. So this is all about the information transfer, how this DNA is copying the information, or this RNA is copying the information from DNA, and this mRNA is translating the information to form the different types of amino acids. So this is all about the story of protein synthesis or gene expression. So with this part your information transfer process unit chapter is over.